Hello, welcome back to part 3 of our look at blood. Again, blood as a tissue. And in this video, we're going to focus all of our attention really on red blood cells, erythrocytes, and how they are produced. In other words, what is the process that we go ahead and create new red blood cells? Red blood cells don't just pop out as red blood cells fully functioning. There is a journey of cellular differentiation that these cells go through. And to start the process, understand that this is a process that is very much determined by hormones. The hormone that is acting upon the stem cell dictates the type of cell that is being produced. So, for instance, the stem cell that we are focused on, is the pluripotent stem cells. So pluripotent stem cells have the ability to differentiate into a wide variety of cell types. Those cell types include even osteoprogenitor cells. So pluripotent stem cells can actually differentiate into osteoprogenitor cells, which is the main cell type found within the skeletal system. But for our look here, for our attention here, those pluripotent stem cells will differentiate into what we could define as being myeloid cells or lymphoid cells. The reason why that is important is because when we look at what those cells give rise to, those pluripotent stem cells give rise to things such as erythrocytes, neutrophils, eosinophils, basophils, platelets. And so these myeloid cells really have the ability to differentiate into a wide variety of formed elements. Again, our look here is on the formation of red blood cells, but all of those cells are going to be derived from myeloid cells. And then the lymphoid cells, as you may have potentially guessed, those lymphoid cells are going to be your lymphocytes. All right. And the other thing that's over here are under myeloid cells that I forgot to mention are the monocytes as well. So when we talk about stem cells, those pluripotent stem cells are critical in the formation of all of our formed elements. I will reiterate the idea that what determines the formed element to be produced is directly linked to the hormone that is acting upon the pluripotent stem cell. Right? So in this scenario, to create a red blood cell, to create a fully functioning erythrocyte, the pluripotent stem cell is going to be acted upon by erythropoietin, which we've already talked about is released from the kidney. Right? And from there, the pluripotent stem cell differentiates into a myeloid cell, and that myeloid cell, under the direction of erythropoietin, is going to differentiate into a progenitor cell. Right? And the progenitor cell will differentiate into a precursor cell, for which there are two forms. There's really like an early precursor and a late precursor. The early precursor cell is a proerythroblast, And the prourethroblast is highly mitotic. In other words, it undergoes rapid cellular division, and it's also within the prourethroblast stage that hemoglobin is produced and added to the cell. Now, towards the end of being a prourethroblast, so late prourethroblast stage, the nucleus is ejected. In other words, the prourethroblast loses its nucleus and at that point in time becomes a reticulocyte. And the reticulocyte is, again, still a precursor cell. We're not a erythroblast or a erythrocyte yet. And the reason for that is because that reticulocyte has to undergo some anatomical changes. In other words, the reticulocyte develops and becomes biconcave. And so we see that very characteristic shape of a red blood cell. And so that reticulocyte becomes biconcave, right, where the red blood cell from the side looks like this. And so you get a thinning in the middle. And this is because, again, there's no nucleus that's within here. This losing of the nucleus and becoming biconcave actually increases surface area, which means you can have an increase in hemoglobin, which means an increase in O2 capacity. Now, when you look at a red blood cell in a microscope and you're looking at it from a superior or an inferior view, or a posterior, inferior or superior view, you'll notice that it almost looks like a little donut. And in fact, it's not a donut. That's not a hollow cell. What you're seeing is that thinning right here in the center, where that cell is now a biconcave structure. And so the reticulocyte loses the nucleus. It keeps some mitochondria. Right? So in other words, we're able to do cell respiration, right? Because we've got to be able to go ahead and still create some ATP. Ribosomes are maintained. There are some, let me rephrase that. Ribosomes, endoplasmic reticulum, they are released from the cell as well. So we're getting rid of the nucleus. We're getting rid of endoplasmic reticulum. We're getting rid of ribosomes. And what'll end up happening is after about 2 days of development, that reticulocyte is released into the bloodstream. And as it's released into the bloodstream, it becomes a mature red blood cell. And so usually about 2 days after release, we've got a fully functioning, mature red blood cell. There is advantages to releasing these reticulocytes prematurely. In other words, not fully functioning as a red blood cell. We see in some parasitic infections, we see a higher than normal reticulocyte to erythrocyte ratio. And the reason for that is because the body would hope that maybe the parasite or the virus would attack the immature cells instead of attacking the fully functioning red blood cells or erythrocytes. And so sometimes in the case of parasitic infections, viral infections, we see a higher ratio of reticulocytes to erythrocytes in hopes that it will confuse, if you would, the pathogen. All right. And so this here is the progression that we see within red blood cells. And I'm going to just kind of fast forward here real quick. This slide's a little bit out of order, if you would, but here's that pathway. Right? Here is that pathway. So again, pluripotent stem cell. Is going to be acted upon by erythropoietin, causing differentiation into a myeloid cell. Colony forming units. This here is a hormone. This here is a what we call a cytokine. Is going to then go ahead and act upon that myeloid cell to begin that process of creating the prourethroblast down into and becoming then the precursor cells. All right, so again, you've got the prourethroblast. All right, loses the nucleus, becomes the reticulocyte, becomes biconcave, is released into the blood, and usually, typically about 2 days after that, is a fully functioning reticulocyte. What's interesting about this particular diagram is we see that differentiation of what the myeloid cells direct the production of. Again, red blood cells, platelets, eosinophils, basophils, neutrophils, monocytes, mast cells, and then we see what the myeloid cells become, which are all lymphocytes. T cells, B cells, and NK cells are natural killer cells. I do want to highlight for you that what each of the hormones are that are regulating this process. And so, once again, just to make sure that we are all on the same page, all right, red blood cells are going to be under the direction of erythropoietin. And erythropoietin, remember, is released from the kidneys. All right, platelets. Platelets are under the direction of thrombopoietin. And remember that thrombopoietin is secreted from within the liver. And so thrombopoietin acts upon the pluripotent stem cells to go ahead and cause differentiation, where now we have colony-forming units, megakaryocyte enzymes that drive that differentiation of and formation of the platelets. We also have, for our leukocytes, our white blood cells, we have a couple different things going on here. We have leukopoietin, that is being produced and released within the bone marrow. To help drive the formation of the white blood cells. But this is where things get a little bit of interesting, because this guy right here, the neutrophils are also what we refer to as being autocrines. and paracrines. And so what that means is neutrophils have the ability to release these cytokines, again, interleukins, that can self-stimulate the production of more neutrophils, hence the term autocrine, and they can actually stimulate through the release of interleukins other white blood cells to proliferate and divide, what we would define as being a paracrine effect. So, and monocytes have the same ability as well as a macrophage. And so if it's noticing a high rate of inflammation, it can stimulate other white blood cells to become differentiated, to divide, to go ahead and support whatever the pathogen may be that is causing the infection. And so, again, by definition, a cytokine is a protein that stimulates an immune response These cytokines can be released from neutrophils and monocytes specifically. And we're going to talk more about this when we get into the immune system as well. But examples of cytokines include interleukins. Remember, there are 40 different types of interleukins that our white blood cells can secrete. All right. Another protein called an interferon. We'll talk more about these in a little bit. We also have those CSFs, those colony stimulating factors. And so neutrophils can specifically secrete CFU erythrocytes or CFU megakaryocytes or CFU granulocyte enzymes to go ahead and cause that differentiation of those myeloid stem cells. All right. Now, remember that we also talked about the fact that cytokines can function one of two ways. They can be a autocrine, which means the immune cell can secrete an interleukin or an interferon or a CSF, and it can act upon itself. So it's got receptors that the cytokines are binding to that will cause proliferation by itself. Or it can have a paracrine effect, meaning that the interleukin or the interferon or the CSF unit can be released by something such as a neutrophil, and it can stimulate the production of other myeloid-based stem cells. The power of the neutrophils also extend even into the lymphoid stem cells. So neutrophils can secrete interleukins that will stimulate T cells and B cells to also begin replicating themselves and becoming divisible to go ahead and increase their numbers. And that is unique in the fact that you've got a myeloid cell technically now regulating through paracrine function a lymphoid cell. But remember that your lymphoid cells, your T cells and your B cells, are adaptive immune cells. And so they're specific for a targeted antigen on a pathogen. And so you got a lot of versatility within our neutrophils. Neutrophils are fascinating if you really sit down and you start looking at them, um, and you really start studying them. They are by far in control of your immune system. They can again regulate this entire process, all based off of the secretion and the release of these cytokines. Um, uh, just to kind of give you a little bit of a background, interleukin 4, interleukin 5, interleukin 13 um, will go ahead and aid in the differentiation of B cells. And so they lead to B cell differentiation. Interleukin 1 will go ahead and activate lymphocytes to go ahead and secrete interleukin 2. Interleukin 2 is a autocrine that will stimulate T cells. And so interleukin 1 stimulates lymphocytes, T cells and B cells, to secrete interleukin 2. Interleukin 2 stimulates T cells that are nearby. And so they have both a autocrine and a paracrine effect. Interleukin 8 is produced by monocytes and fibroblasts, and targets neutrophils and basophils and macrophages and mast cells to undergo what we call chemotaxis. And so neutrophils actually have the ability to destroy through a killing zone. It's a gaseous zone. They can kill and destroy other um, um, pathogens. But interleukin 8 can also stimulate angiogenesis, which is the rebuilding of new blood vessels. And so if you've got a damaged area that needs new vascularization, interleukin 8 will stimulate that angiogenesis, that production of new blood vessels. It will also cause the release of what we call superoxide, and superoxide is what creates um, is what aids in the creation of, sorry about that, hydrogen peroxide, which will go ahead and aid in the destruction of other pathogens, and also the release of other interleukins. And so interleukin 8 has a lot of versatility as far as repair within specific tissues. You get a cut, or you get ink work done and you're getting new ink added onto a tattoo. Your cells are actively secreting, your monocytes and your fibroblasts are actively secreting interleukin 8 to go ahead and manage that inflammatory process, which in turn, believe it or not, kind of embeds the ink a little bit more into the skin. So fascinating, once again, fascinating regulation and control that we find within these interleukins, and we find woven into the neutrophils. I could spend another 30 or 40 minutes going over that, but I will not do that to you, because we have a lot of other things I kind of want to also get to with this. There's the pathway. We kind of already looked at that, so I'm not going to spend any more time on that. This kind of branches, or at least removes kind of this view of that diagram there, and kind of isolates it out for you. All right. We've already talked about some of the hormonal controls, so this is a review for us, if we would, all right, based off of what we've already looked at. And so that should be okay. And really, the structure of our red blood cells, this is not something that is probably new to you. And so a lot of this is probably a review. We know that red blood cells, erythrocytes, have hemoglobin. We know that the role of hemoglobin is to carry oxygen. All right, oxygen binds to the heme group of the hemoglobin. So the globulin is the protein. The heme, is the protein that has an iron molecule, and that is where the oxygen binds to. We know that red blood cells have a biconcave structure, that it lacks a nucleus, it lacks most organelles, and because of that, it cannot reproduce on its own. Um, and that hemoglobin is really maintained within the cytoplasm, what we call the cytosol, within the red blood cell. And so a lot of this we've already talked about. I will add to this, just because I like adding little bits of information to drive you guys crazy. When we talk about ATP production, remember, mitochondria is maintained. Right? And so ATP production within the mitochondria is actually happening anaerobically. And so it's not going to use the oxygen that it's carrying to conduct its own ATP production. It's going to do this anaerobically to conserve the oxygen. Again, less efficient, right, than doing it aerobically, but that's okay because there's less metabolism going on within the red blood cells. And so that's perfectly acceptable to do that. The average lifespan of a red blood cell is 120 days. Right, so you're dealing with about 4 months that a red blood cell will be in existence. And because of this increased surface area, due to all of this, right, again, we kind of already mentioned this, on average, right, on average, each red blood cell will contain approximately 280 million hemoglobin molecules, HB. And if each of those hemoglobin molecules can carry 4 oxygen molecules, then that means that we have 1,120 million O2 molecules per red blood cell that is being carried. All right. And so that is a phenomenal amount of oxygen that each red blood cell can go ahead and carry. And here is the structure, the overall structure of hemoglobin. All right, so we see the globulin part is made up of 4 globular proteins, 4 tertiary chains, or I should say 4 tertiary globular proteins that join into one quaternary structure, right? 2 alpha chains and 2 beta chains. And that on each one of these chains, we have that heme group. And at the middle of that heme group is the iron molecule. And that is where the oxygen is binding to. And so I want to stop here for this video because the next couple of things, the next 2, 3, the next 3 slides, 4 slides are very detailed and in-depth diagrams, and so I want to spend some time on that on a separate video. So we're going to stop here, give you some time to digest all of this hormonal regulation that is going on with the production of our erythrocytes as well as our leukocytes. Again, give you an opportunity to kind of jot down some questions and shoot me an email or ask me during our Q&A sessions, our little class time sessions, questions that you may have. And so with that said, get reviewing, get studying, think of those questions, what's not making sense, and I'll catch you on the flip side.